Hi, my name is Caroline Pitts, and today we're going to talk about transcription in a prokaryote. Now, before we begin, make sure you familiarize yourself with a couple of these vocab terms. Transcription is the process by which the information in a strand of DNA is copied to a new molecule of messenger RNA, and RNA is used to produce protein in cells. Now, to begin, the 4 stages of transcription are template binding, chain initiation, chain elongation, and chain termination. RNA is transcribed from a single strand of DNA in the uncoiled section of a double helix structure, with our template strand serving as a template for transcription and is read 3 to 5 prime. Our coding strand, being our opposite strand, is antiparallel and complementary to the DNA template, which sequence corresponds to the RNA being synthesized that we translate into protein, being read 3 to 5 prime. Here we can see how complementary nucleotides work within this process. If we simplify this, we can see that transcription begins at a place called the promoter region. The promoter region includes the -35 site, the -10 box or TATA box, and the +1 site, which is also known as the initiation site. The RNA coding region also includes the +1 site, but this is where the transcription takes place and is terminated. Additionally, the -35 site and -10 box are what make up the consensus sequence inside the promoter region. The TATA box is where part of the DNA sequences begin to decode, and it's called the TATA site because it only includes thymine and adenine. Next, we add a core enzyme and sigma factor. The RNA polymerase gives us the core enzyme, which is capable of synthesizing RNA, and the sigma factor enables the core enzyme of RNA polymerase to attach the coding strand in the promoter region and create a hollow enzyme. Next, RNA polymerase binds to a consensus region and begins to produce RNTPs, and then the process of termination begins. Row-independent termination must include specific sequences of G and C template strands, which causes the RNA transcript to fold on itself. The RNA strand creates a hairpin loop due to inverted repeats and needs to have all the right complementary base pairs. The hairpin, as well as all the things around the hairpin, impair the RNA polymerase, and transcription is paused. Additionally, the complementary UA region causes a weakened interaction, leading to enzymes falling off, and the new transcript is freed. Hypothetically, if there is a 5 BP insertion between the -35 region and the -10 box of a gene that encodes for an enzyme required for the production of the amino acid phenylalanine, what would be some of the consequences? With the insertion causing a mutation in the promoter region, the sigma factor would likely be unable to recognize where it is, and the RNA polymerase would not be able to bind to the DNA, leading to an inability for transcription to occur.